right, the last chapter that pertains to our first module is chapter 4, and we continue to look at details within the cell, specifically the genetic material, which is pretty much the most important thing that the cell has. It has genetic material that is composed of DNA. DNA stands for deoxyribonucleic acid. This is a long thread-like molecule. You may be familiar with it as a double helix. You can see that here on the right. It has a uniform diameter, but it can vary in length. We have 46 DNA molecules or chromosomes in the nucleus of most cells. These cells are called somatic cells. The only time we don't have 46 is when we're talking about the gametes. And the gametes only have 23 chromosomes, one gamete being the egg and one gamete being the sperm. So when those two combine, they give the resulting zygote 46 chromosomes. So what exactly is DNA and how does it work? DNA, or deoxyribonucleic acid, and other nucleic acids, the only one that you'll really need to be familiar with in this class is RNA, which is ribonucleic acid. Both of these nucleic acids are polymers of nucleotides. And so each nucleotide contains a sugar, either deoxyribose in the instance of DNA, ribose in the instance of ribonucleic acid or RNA, a phosphate group, and then both of these are attached, specifically the sugar is then going to be attached to a nitrogenous base. Here in the figure we have the nitrogenous base adenine. There are 4 main nitrogenous bases in DNA. They are A, T, C, and G, or adenine, guanine, cytosine, and thymine. This 5th one listed here, uracil, is specific to RNA. And DNA does not have uracil, but RNA does not have thymine either, and we will talk about this more in a minute. Your book combines these nitrogenous bases based on their shape. I'm not as concerned with you knowing what category they fall under, but for your information, purines are going to be the double ringed structures that are adenine and guanine, and the pyrimidines are going to be single ring structures, cytosine, thymine, and uracil. I'm more concerned with you knowing the names, adenine, guanine, cytosine, thymine, and uracil, and which structures they belong to. The DNA structure, again, is a double helix, like we see here on the left. And that double helix is actually a twisted pair of single-stranded nucleotides. And so it ropes around like a 3-dimensional shape. The double strands, or the 2 strands of nucleotides, are basically going to have a backbone. So here are 2 right here, 2 backbones of alternating phosphates and deoxyribose. And then that deoxyribose is attached to the nitrogenous base. The purines are going to pair with pyrimidines, so we have specific pairings that you need to learn. A will always pair with T and G with C, and vice versa. So T would pair with A, as you see here, and C will pair with G. This is called the law of complementary base pairing. Essentially, because we know these nitrogenous bases will always pair with a certain other one, then we can determine what a complementary strand would be. So if we know the sequence of one strand, we'll know what the complementary base will be. So for instance, if we were only given this strand here on the left, and it has A, G, T, and G, we would know without looking at the second one that it would have to be a T. It would have to be pairing to a T, a C, an A, and another C. Simply because of what nitrogenous bases pair with each other. What is the function of DNA? It is going to hold instructions that we call genes to synthesize specific proteins. A lot of times what I like to call it is it's like a recipe book. It's a recipe book to make a certain recipe or a certain food dish. And the food dish in this case is the proteins. And we need proteins for essentially everything we do and to help make us. So a gene is just a segment of a DNA sequence, something like what we have right here, but in a lot longer or a lot more nitrogenous bases in a row, that's going to encode the directions for how to make a specific protein.